Accumulation of variation during reproduction. Reproductive processes give rise to new individuals that are similar but subtly different. If we observe a field of sugar cane, we find very little variations among the individual plants. However, in number of animals, including human beings, which reproduce sexually, quite distinct variations are visible among the different individuals. In case of asexual reproduction, new generation reproduces and the resultant two progeny divide again. The four individuals generated would be very similar. There would be only very minor differences among them generated due to small inaccuracies in the DNA copying. However, in sexual reproduction, greater diversity will be generated. But what is the probability that these variations in a species have equal chances of surviving? For example, a bacterium that can withstand heat will survive better in a heat wave. Selection of variants by environmental factors forms the basis of evolutionary processes. Inherited traits. Heredity is the continuity of characters from one generation to another. It is present in the DNA of the fertilized egg or zygote. The rules of heredity determine the process by which traits and characteristics are reliably inherited. Let us take a closer look at these rules. The progeny inherit the traits from the parents. These traits are physical features like hair color, height, skin color and nose. You appear similar to your parents because you have inherited these traits. Let us now study the rules for the inheritance of traits and Mendel's contributions. Mendel worked out the main rules of such inheritance, and it is interesting to look at some of his experiments. Mendel carried out his experiments by using garden pea with the 7 contrasting visible characters. They are flower color, violet and white flowers, flower position, axil or terminal, seed color, yellow and green, seed shape, round and wrinkled seeds, pod shape, inflated and constricted, pod color, green and yellow, and stem length, tall or short. Mendel took a tall plant and a short plant, produced progeny from them, and calculated the percentages of tall or short progeny. Capital T are tall plants and even capital T are tall, but small t are considered short plants. That is, even if one capital T is present, the plant will be tall. Capital T is dominant while small t is recessive. All plants were tall. This meant that only one of the parental traits was seen, not some mixture of two. The F1 tall plants were self-pollinated, and F2 progeny of the F1 tall plants are not at all. 3 of them were tall and 1 short. This indicates that both the tallness and shortness traits were inherited in the F1 plants, but only the tallness trait was expressed. Expression of traits. How does the mechanism of heredity works? Cellular DNA is the information source for making proteins in the cell. A section of DNA that provides information for one protein is called the gene of that protein. How do proteins control the characteristics that we are discussing here? Mendel took 2 characters at a time and crossed them. This is a dihybrid cross. A dihybrid cross is a cross between 2 parents that breed true for different versions of 2 traits. In this example, the flower color is governed by A and plant height by B. Tall and purple flowers are dominant over the dwarf and white flowers. Tall and purple colored plants, capital AABB, will produce gametes capital AB, 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 AB. While dwarf and white colored plants, small AABB, will produce gametes small AB, 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 AB. 9 out of them will be purple and tall, 3 would be purple and dwarf, 3 of them will be white and tall, and only 1 will be white and dwarf. The ratio will be 9:3:3:1. If the progeny plants inherit a single whole gene set from each parent, then this experiment cannot work. This is because the two characteristics capital A and small b would be linked to each other and cannot be independently inherited. Each gene set is present not as a single long thread of DNA but as a separate independent pieces, each called as chromosome. Thus, each cell will have two copies of each chromosome, one each from the male and female parents. When two germ cells combine, they restore the normal number of chromosomes in the progeny, ensuring the sustainability of DNA of the species. Such a mechanism of inheritance explains the results of the Mendel experiments and is used by all sexually reproducing organisms. How are these genes governed? For example, the tallness of a plant is dependent on the amount of a particular plant hormone. Now consider an enzyme that is important for this process. If these enzymes work efficiently, a lot of hormones will be made and the plant will be tall. If the gene of that enzyme has an alteration that makes the enzymes less efficient, the amount of hormones will be less and the plant will be short. Sex determination. How is the sex of a newborn individual determined? There are different criteria of determining the sex. In case of some animals, the temperature at which the fertilized eggs are kept determines whether the embryo developing in the eggs will be male or female. Snails' individuals can change sex, indicating that sex is not genetically determined. In human beings, the sex of an individual is largely genetically determined. All human chromosomes are not paired. Most human chromosomes have a maternal and parental copy, and we have 22 pairs of autosomes and 1 pair of sex chromosomes. Women have a perfect pair of sex chromosomes, both called as X. So women are XX. But men have a mismatched pair in which one is a normal sized X while the other is a short one called Y. Therefore men are XY. All children will inherit an X chromosome from their mother, whether they are boys or girls. Thus the sex of the children will be determined by what they inherit from their father. A child who inherits an X chromosome from her father will be a girl, and the one who inherits a Y chromosome from him will be a boy. Evolution. Consider a group of 12 red beetles that live in bushes with green leaves. Their population will grow by sexual reproduction and therefore can generate variations. Suppose the crow eats the beetles. The more beetles the crow eats, the fewer beetles are available to reproduce. Case 1. A color variation arises during reproduction, so that there is one beetle that is green in color instead of red. This beetle moreover can pass the color, so all its progeny beetles are green. Crows cannot see the green colored beetles on the green leaves of the bushes and therefore cannot eat them. The progeny of green beetles is not eaten, while the progeny of red beetles continues to be eaten. As a result, there are more and more green beetles than the red ones in the beetle population. Case 2. Again a color variation arises during reproduction, but now it results in a beetle that is blue in color. This beetle can pass the color on to its progeny so that all its progeny beetles are blue. Crows can see the blue colored beetles in the green leaves of the bushes as well as they can see the red ones and therefore can eat them. In the population as it expands, there are a few blue beetles but most are red. But at this point an elephant comes by and stamps on the bushes where the beetles live. This kills most of the beetles. By chance the few beetles that have survived are mostly blue. The beetle population slowly expands again, but now the beetles in the population are mostly blue. In both cases, what started out as a rare variation came to be a common characteristic in the population. In other words, the frequency of an inherited trait changed over generations. Now consider the case 3. In this case, as the beetle population begins to expand, the bushes start suffering from a plant disease. The average weight of adult beetles decreases, but there is no genetic change occurring. After a few beetle generations, the plant disease is eliminated. At this time, what would we expect the weight of the beetles to be? The beetles will grow in normal size. Acquired and inherited traits. Traits are the qualities or features that distinguish an organism, like the hair color, height, skin color, voice, and so on. However, the traits can be acquired traits and inherited traits. The acquired traits cannot be passed on genetically, while the inherited traits can be passed on to the next generation. For example, if we breed a group of mice, all their progeny will have tails as expected. Now, if the tails of these mice are cut in each generation for 20 generations, do these tailless mice have tailless progeny? The answer is no. And it makes sense because the removal of the tail cannot change the genes of the germ cells of the mice. Charles Darwin theory of evolution. Charles Darwin conducted various experiments which led him to formulate his hypothesis that evolution took place due to natural selection, the struggle for existence, and the survival of the fittest. Darwin's theory of evolution tells us how life evolved from simple to more complex forms. The first primitive organisms would arise from further chemical synthesis. Experiment conducted by Stanley L. Myler and Harold C. Urey in 1953. They assembled an atmosphere similar to that thought to exist on Earth only. This had molecules like ammonia, methane, hydrogen gas and nitrogen, but no oxygen over water. This was maintained at a temperature just below 100 degrees Celsius, and sparks were passed through the mixture of gases to stimulate lighting. At the end of a week, 15% of carbon from methane had been converted to simple compounds of carbon, including amino acids, which makes up the protein molecules. Speciation. Speciation occurs when one species splits into two species which later evolve into distinct lineages. There are three kinds of barriers for gene flow. They are allopatric, sympatric, and parapatric. Allopatric speciation occurs when geographic barriers prevent the gene flow between populations. They will develop new traits and eventually won't be able to interbreed. Sympatric speciation occurs when species gain increase in the number of chromosomes. Eventually, the germ cells of the two groups cannot fuse with each other, or a new variation emerges in which species A females will not mate with species Z males, but only with species A males. Parapatric speciation. This time the populations are not isolated by a physical barrier, but are instead beside each other. Individuals within the population only mate with individuals in their immediate area and produce a hybrid zone. Evolution and classification. Characteristics decide more fundamental differences among organisms. A basic characteristic of cell design that differs among different organisms is whether the cell has a nucleus. Bacterial cells do not have a nucleus, while the cells of most other organisms have. Among organisms with nucleated cells, which ones are unicellular and which ones are multicellular, this property marks a very basic difference in the body design because of specialization of cell types and tissues. Among multicellular organisms, whether they can undertake photosynthesis or not will provide the next level of classification. Among the multicellular organisms that cannot do photosynthesis, whether the skeleton is inside the body or around the body will mark another fundamental design difference. Tracing evolutionary relationships. The basic structure of the limbs is similar, though it has been modified to perform different functions in various vertebrates. For example, limbs of the frog, lizard, human and bird appear similar but do not perform similar functions. They are called homologous organs. Let us look at the wings of birds and bats more closely. We find that the wings of bats are skin folds stretched mainly between the elongated fingers, but the wings of birds are a feathery covering all along the arms. They look similar because they have a common use for flying, but their origins are not common. This makes them analogous characteristics. Fossils and evolution by stages. Fossils. What are fossils? When organisms die, their bodies decompose and are lost, but every once in a while, the body or at least some parts may be in an environment that does not let it decompose completely. For example, if a dead organism gets caught in hot mud, it will not decompose quickly and the mud will eventually harden and retain the impression of the body parts of the insect. All such preserved traces of living organisms are called fossils. Link of fossils in the process of evolution. Look at these pictures. These are the fossils of 4 different organisms, while this is a fossil of a dinosaur which had feather imprints. This indicates that some dinosaur had feather but would not be used for flight. These were gradually adapted by birds who used them for flying. Hence it is clear that the birds evolved from dinosaurs which were reptiles. Thus we conclude that fossils provide evolutionary relationship between different species. Evolution by stages. The eye seems to be a very popular adaptation. Planaria cockroaches do have them, so does an octopus, and so do vertebrates. The structure of the eye in each of these organisms is different enough for them to have separate evolutionary origins. Humans have over more than 2000 years cultivated wild cabbage as a food plant and generated different vegetables from it by artificial selection. To select for very short distances between leaves and have bred the cabbage we eat. Some wanted to select for arrested flower development and have bred broccoli, or for sterile flowers and made the cauliflower. Some selected for the swollen parts and came up with kohlrabi. Some simply looked for slightly larger leaves and came up with leafy vegetable called kale. Molecular phylogeny. Changes in the DNA during cell division would lead to changes in the proteins that are made from this new DNA. These changes would accumulate from one generation to the next. The organisms which are more distantly related will accumulate a greater number of differences in their DNA. Evolution and progress. It is not as if one species is eliminated to give rise to a new one. There are multiple branches possible at each and every stage of this process. A new species has emerged, the old species will disappear. It will all depend on the environment. Both human beings and chimpanzees have a common ancestor a long time ago. That common ancestor is likely to have been neither human nor chimpanzee. Evolution is simply the generation of diversity and the shaping of the diversity by environmental selection. Human evolution. The following have been used for studying human evolution: excavating, studying fossils, and determining DNA sequences. Regardless of where we have lived for past few thousand years, we all come from Africa. The earliest members of the human species, Homo sapiens, can be traced there. Our genetic footprints can be traced back to our African roots. A couple of 100,000 years ago, some of our ancestors left Africa while others stayed there. The residents spread across Africa. The migrants slowly spread across the planet from Africa to West Asia, then to Central Asia, Eurasia, South Asia and East Asia. They traveled down the islands of Indonesia and the Philippines to Australia and then crossed the Bering Land Bridge to the Americas. Like all other species on the planet, they had come into being as an accident of evolution.